To introduce John Bryant again, who is Professor Emeritus of Cell and Molecular Biology at the University of Exeter. He has the distinction of introducing one of the very first bioethics courses on biology students in a UK university. And he's run workshops for biology academics on teaching ethics. Amongst his distinctions, he's a past president of the Society for Experimental Biology. He's given the London Lectures of Contemporary Christianity, and he's just recently stopped being Chair of Christians in Science, though with the great achievement in his final couple of years of getting charitable status for the Society, which is tremendous. It helps a lot. He's a member of the International Society for Science and Religion, and carried out research on the relationship between Christian belief and attitudes to bioethical issues. So I don't think we have anyone better qualified to talk about this topic, the ethics of stem cell research and training.
Well, thank you very much, Rodney, for the kind introduction. I can think of plenty of people more qualified than I to talk about this. Let me say before I start that there is an emailable version of the PowerPoint presentations for both these talks. I think it's in your hands yet. So
yes.
If you want these slides, it won't have all of them in, then please see Pauline.
They're about 3 or 4 megabytes, or?
Well, yeah, at least I should think. The long versions, you could almost hear them going clunk, clunk, clunk. Okay, we're staying with biological research, and we're staying with my plea that we think about and pray about folk who are involved both in research, and there are some Christians involved in this research, and those who are making ethical decisions about it. One such person is the former head of the Human Fertilisation Embryology Authority. She's now head of the Charities Commission, my friend Susie Leather, who is a Christian and thought deeply about Christian aspects of all the embryonic research for which the HFEA is responsible. So, we're going to do some science as well as looking at the ethics, as we did for the genetics. Come on. Now, I want to introduce the context in which we're talking about stem cells, because it's been a particularly interesting example of how an agenda can be driven by particular interest. This is, you probably recognise, is an ageing Bob Dylan, of whom I'm a great enthusiast.
Yes.
But in one of his weaker albums that he made in the 1980s, he talked about living in a political world where decisions that affect human beings were made not for reasons of love and care, but for political expediency. We live in a political world. And we'll see that there's an element of this as we think about stem cell research. We're living in a political world. And we also live in a highly secular world, in Britain anyway. Maybe it doesn't apply to other countries in which stem cell research goes on, but this was a quote in The Guardian from about this time last year, and we've seen the development of that in Alan's talk this morning, kind of aggressive secularisation of British society. And of course we've also got postmodernism to cope with, the fact that, denying the possibility there are any big stories, such as the possibility of a creative God with overarching care for his universe, and again, quote from Dylan, you're right from your side, I'm right from mine. Perfect example of postmodern approach to morality. This was to do with relationships, so the song. All that affects our thinking about the ethics of modern biomedical research. So let's go back to the early human embryo. As I said before, this is the fertilized egg. One sperm has got in. It takes about 100,000 to mount the cavalry charge to get in there, as you've seen in the Woody Allen film, as they go over the top, and it goes through these series of divisions, as in nature, this tiny structure, so 0.1 of a millimetre. The size hardly changes as these cells divide, because this cell is so big, and so you're reducing the cell size, hardly changing the overall size of the embryo. Until we get, at about 7 days in nature, to this structure here, which has between 60 odd and 100 odd cells. The number is sort of indeterminate, but it's like a little tiny football. It's hollow, and it has a layer of cells on the outside, and it has a little mass of cells hanging from the inside of the skin. So if you imagine you could slice through a football without it losing its kind of its integrity, you'd see inside, hanging in the skin, a little ball of cells. That's called the inner cell mass. And this structure is called the blastocyst. And at this stage of embryonic development, the woman is still not pregnant. This is all happening with a free living, as it were, embryo, though supported by the nutritional environment of the fallopian tube. The woman is still not pregnant. What will happen if a pregnancy is to be established is that the blastocysts, as it were, hatch out, and these cells will break out, and they will implant on the wall of the uterus. And the outer, and this will become the embryo and then the fetus. And the outer layer will become the placenta. So by this time, we're beginning to see some sort of division of labour. Placenta, and this will become the embryo proper, and then it's the fetus. And it's only after that happens we begin to see any polarity, any front, back, up, down, left, and right. We still haven't got any polarity there. So the key thing to note then is that that little structure there, the inner cell mass of cells, is going to become the embryo. And if you think about what happens in embryonic development, we first get the embryo dividing into approximately three layers, well, actually 3 layers, which is more or less well defined. Each of those 3 layers has got particular function in the formation of subsequent tissues and organs. So one layer will form skeleton, for example, another layer will form the nervous system. But eventually, the fetus and then the baby will have about 210 to 220 different sorts of cell gathered together in tissues, gathered together in organs. Something like 210, 220 different sorts of cell. But these cells here have the potential to make them all. They must do, otherwise it would not happen. So this inner cell mass is what is called a stem cell population. So if we go back one, that's the zygote, the one-cell embryo. It comes from a Greek word, zygosis, coming together. That's got the genetic potential to make everything, placenta and embryo. And so it's got complete potential for development of life. And then we get that division of labour. Although the cells that become the placenta have still got all the genes, and although the cells that become the embryo have still got all the genes, they began to divide in terms of their function. But when we look at what's happening to genes in that period, as we go from the blastocyst to the fetus to the baby, it is quite extraordinary what's going on there. That we are seeing switching on and off of individual genes, of great blocks of genes. We're seeing interactions between individual genes and great blocks of genes. We're seeing responses to signaling coming from the position the embryo is located on the womb. We're seeing nutritional signals coming from the mother. So we see a huge array of switching mechanisms going on, switching genes on and off. And I always liken it. I was at a gig the other day in Plymouth with the rock band, and this was actually behind the techies' desks. They had 3 desks and 3 computers, and 2 techies working, 2 roadies working the whole show. It looked very complicated. But that complexity has got nothing on what goes on in running a human being, starting off a human being with its gene switches from day 1. So here we are again, going through this, and as I say, this will establish a pregnancy, and then 9 months later you get one of these. Not one of these. That's Daniel, he was 3 last week. And so we get to the idea then, if we go back to that blastocyst, that little group of cells, will end up producing one of these, all its different cell types. So they are cells which have got this enormous potential. And we call them stem cells. Stem cells have got 2 features. One is that to some extent they will self-renew, so cell division will produce more of them. And secondly, that from a stem cell population, we will get a number of different other cells. And obviously, if we're talking about those stem cells in the embryo, it's not just that they will give many different cell types, they can give rise to all the cell types in the born human being. all 210, 220. So they, to some extent, they self-renew, and they produce different types of cell. And it's been suggested that stem cells might be used in medicine to repair damaged tissue or damaged organs that have been damaged by illness or by trauma, such as Christopher Reeve, former Superman, who died eventually of his injuries, but he had a high break in the spinal column. Maybe it's just old age that is causing degenerative illness. But the idea is you might be able to repair damaged tissues and cells by using stem cells. And of course, if we're looking for the most potential, if we're looking for the cells that have got the most potential, because they will lead to the formation of 200 plus cell types, if we're looking for the cells with the most potential, then the blastocyst is the obvious place to look, a source of embryonic stem cells. We're going to come to other sorts of stem cells later. And so this is a kind of cartoon about what stem cells do. So I want you to imagine that little point there is a cell division, and it's given rise to one more stem cell, and it's given rise to a cell that is on its way down a developmental pathway. And that will divide again and again. So we see stem cell population being retained, and this is turning in this simple cartoon into two different cell types. And so here's the idea then, that maybe we can use the cells in the blastocyst, in the human embryo. We can take those out, we can culture them as blastocyst cells, and obviously we maintain the population, but maybe we can induce them to go down this pathway, and then that pathway, down this pathway, and down that pathway. In other words, can we, in the lab, induce the developmental programs that we normally see in nature? And that would be the idea of using embryonic stem cells. So this is how it works, that you take the stem cells out of the embryo, and I'm at this point avoiding ethical discussion, that will come later. You take the stem cells out of the embryo of course no longer will develop into a human being. You keep them in the lab, and we immediately run into a problem. I said stem cell populations self-renew, and as we'll see later, the ones in the human adult body do self-renew, but the stem cell phase in an embryo is quite short, a couple of days or so, it's quite short. And so the first problem you have is to keep themselves in that youthful state when they're going on maintaining themselves. You've got to keep them in that embryonic stem cell state. People say that scientists have got no soul, but here we go. A gene that keeps them in the youthful state was discovered in Edinburgh by a Gaelic-speaking scientist, and he used the Celtic word for of youth, nanoch of youth. And it's the same in Irish, and it's the same in Breton. So there's the gene of youth, and if you can keep that gene active, then you keep the cells in that constantly dividing stem cell state. And then what you do is that you take some of those cells and you try to push them down particular developmental pathways. And there has been some success. But remember, normally this is all a regulated process that's occurring in embryonic growth and development. A huge amount of spatial and temporal regulation is going on here. We remove the stem cells from the body. We're trying to persuade them to do in the lab what they would normally do in nature. But there has been quite a lot of success, particularly in Stephen Minger's lab at King's College London. So where do we get the cells from? Well, this was the first source of embryonic stem cells. Human stem cells were first cultured as recently as 1998. The stem cells were discovered in mice long before then by, amongst other people, Martin Evans, who was here in Cambridge at the time, is now in Cardiff, and is, I think he's recently become a Nobel laureate. So he was one of the people who discovered mouse embryonic stem cells and how to culture them. Human embryonic stem cells took a little while longer, and it was first done by the States, actually. But the embryos came from in vitro fertilization, and those of you who know about this will realize that in an average cycle of IVF, between 6 and 10 embryos are made, depending how many eggs the woman produces. And only a very small number of those are used to establish a pregnancy, and the rest are spare, if I can put that in inverted commas, and they're generally stored in liquid nitrogen in the freezer. some of those embryos have been used in this research. All this research is regulated by the Human Fertilisation and Embryology Authority. Any research on embryos, anything to do with manipulating human embryos in Britain, has to be regulated by the HFEA. There's no possibility of working outside their remit. Whereas in the States, it's very different. That there is no overarching regulatory body. That there's a federal ban on working with embryos, but if you get your funding from somewhere else, you can do it. So you have this one strange split situation in the States. And here, for this work, as I say, the license has been granted to King's College at London University under the leadership of Dr. Stephen Minger. Many of you will have seen him on television. You go to his website and you'll see him there with his long ponytail, nicely plaited. One of my students is going down to talk to him next Monday. Okay, let's go back to genes and development. Go back to the zygote, that one cell embryo. And again, a reminder, in that one cell is the genetic potential for making the complete person at the genetic level. We're not talking about the other influences like they were this morning. And again, we have to think about the genes being switched on and off. And one of the things that happens in mammals is that genes that have been switched off and have had their time during development are actually quite hard to switch on again. And so, for example, if you were to look at the genes active in my brain cells, and there are some cells up there, I gather, if you look at those genes, you would find the whole banks of genes switched off because the brain cells do not need the liver enzymes. So all those genes will be switched on. And so we have this complex array of switching on and off. But in some cases, the switching off looks pretty permanent. The genes are there, but they're very hard to switch on again. But if we were to switch them on again, all the genes are there, apart from in the red blood cells. Is it ever possible to get those to start again, to behave as if they were back in the embryo? So all the genes are potentially active. Well, that's effectively the basis of cloning, and we're going to look at this in stages, because this is also got relevance to stem cells. But in cloning, instead of fertilizing this egg, the scientists would take out the egg's genetic material and see whether they could actually use the genetic material from an adult to fire the embryonic development of that egg. That would be a kind of cloning procedure. Now, of course, there are two routes to making clones, one of which I've just described, and we'll look at it in a minute, but I know everybody in this room will know some genetic clones. I can say that without any fear of contradiction whatsoever, because you all know some identical twins. And what's happened with identical twins is that at that very early stage when pregnancy was being established, when that blastocyst was attaching to the wall of the womb, somewhere around that time, the embryo split into two. Sometimes it occurs a bit later and you get conjoined twins. Sometimes it occurs a bit earlier, maybe only one of the embryos actually clings on to the wall of the womb. But around that time of attachment, the embryo can split in half. And we have identical twins. They have the same genome, same genotype. And of course, with monozygotic twins, the clones are the same age. I know that when you meet identical twins, one of them always claims to be older because he or she came out of the womb before there. But actually, they're the same age genetically. Interestingly, and I've never really kind of got to the bottom of this aspect of our law, that you're not allowed to make embryos for IVF in this country and then split them in half to make twins. You're not allowed to do that. You are allowed to do it with cattle. And so an elite embryo in cattle breeding may well be split into up to 4 to make quads in 2 different mothers. But you're not allowed to do it for human embryos. But of course, the other route to clone is the one I've just described, where you take the genetic material from an adult cell and you put it into an empty egg. And we'll see the relevance of this to stem cells in a moment. But of course, what we have here then is that the genetic clone, the two genetic copies are not the same age. One is much younger than the other. And in Dolly's case, Dolly was 6 years younger in time than her mother, although the DNA seemed to behave as if it was still, because of epigenetics and imprinting, still seemed to behave as if it had come from the older sheep, which is a worry. So here's a little cartoon of how it works. This is an egg. Egg is a very big cell. Let's say 0.1 of a millimetre, very, very, very short type people. They can hold it up here, you can probably see it. So the body cell on the same scale would be a dot on this page, and so I've expanded this. And so here's the egg with its nucleus. The nucleus containing the genetic material is taken out, and this cell, which of course contains the required 2 sets of genes, its DNA is put into the egg cell. And of course, its DNA is in this partly switched off state, and it needs to respond to firstly the environment in which it's placed, the egg environment. Secondly, it needs to start dividing, and it's found empirically, but just hit and miss. The best way to start dividing is to give it a very, very short, very high voltage shock. So we're talking microseconds of thousands of volts, and that seems to get the cell dividing and behaving as if it was an embryo. And of course, if the embryo will then carry on to term, then you will get a clone. And so here is Dolly. This is in July 1996. And here's Dolly with her surrogate mother. So they did, in fact, implant the embryo back into the clone donor. So you can see that this is a surrogate, not genetically the same as Dolly. And here she is in February 1997 at her press conference. I don't think a sheep has ever been asked so many questions, ever before or since. But I'm told that she didn't say very much. So, and for those of you who wonder, Harry Griffin at Roslin assures me that she was indeed named after Dolly Parton, which I thought was just an apocryphal story until I talked to Harry. And you may wonder why she was named after Dolly Parton. The reason is that the cell from which the DNA was obtained was from the mammary epithelium. And I'll say no more than that. Now, this story may well be apocryphal, but Harry also told me that when they told Dolly Parton this, and she said, no publicity is bad publicity. But so there's Dolly, named after Dolly Parton, and of course she died young. It was almost as if the DNA had accumulated 6 years' worth of lesions, which were not repairable, even though the DNA started to behave as if it was embryonic. We do not know at this stage how general a phenomenon that will be in cloning. It does seem that we're getting over it, but we don't know. So what's the point of all that? Well, the point of all that is that we're pretty good at manipulating human embryos. We've been doing it in the lab since 1968 through IVF. So would there be any point in doing human cloning? Well, I'm going to leave the reproductive cloning to one side for the moment, but we'll just think about it as firstly difficult. Just to make an embryo this way is difficult, even to get an embryo. The success has been very, very limited. My feeling is that if more of this research was allowed to go on, then it would get better. We would get better at it. But I think I want to emphasize, it must be clear from what I've said, that this is not mass technology. We're not talking about rows and rows of human eggs into which we can inject DNA after DNA after DNA. This is individual manipulation. So again, we're not thinking of the boys from Brazil scenario, where those women have been implanted with clones all over South America to make these convincing neo-Nazi style Hitlers. When somebody asked me, could you have a football team with 11 Ronaldinos in it? And I said, speaking as a wicketkeeper, I identify with goalkeepers. I said, you need a goalie. And they said, no, not with 11 Ronaldinos, you wouldn't need a goalie. But you can't, it's not mass technology. This would be one at a time. This is just an aside. Are there special ethical issues that arise from the ability to make genetic copies of pre-existing human beings? Just something to think about. But let's go back to stem cells, and this is the point of introducing cloning at this time. If we produce an embryo by cloning, and we were trying to repair a person, we could use that person's DNA to make the clone. So, supposing that I had some sort of degenerative condition that wasn't a heritable condition, but a somatic condition and needed repair, idea would be that I would turn up at the clinic, one of my cells would be taken, the DNA would be obtained, an empty egg would be obtained, and there's a big question, where does that come from? And a clone of me will be made. And so any stem cells generated by that route would be compatible with my immunological system. There would be no possibility of rejection. So the idea of therapeutic cloning, as it's so called, is to make tissue grafts which would not be rejected by the recipient. And licenses have been granted to, in this country, or in Great Britain, to Newcastle and to the Roslin Institute, which is the lab which first cloned Dolly. The research is going on in lots of other places. It's going on in the States. As I say, federally, there's a federal ban on federally funded work in this area, but there are companies and charities that are working very actively on human cloning to make stem cells. It's a subject in the States that's even featured in the previous 2 presidential elections now. People have got up and spoken about the need to support this work. It generates a lot of heat. Before he died, Christopher Reeve talked about bigots were delaying his recovery, meaning people that disagree with stem cell research by bigots. So work is going on in the States, but it's not regulated in the same ways over here. So if you looked up, if you Googled cloning USA, you'll probably find many companies that are doing this. It's also happening in Korea, where there's quite a good biomedical research record. And one cloned embryo has been produced in Newcastle, but research has not proceeded. Cloned embryos have been produced in Korea. There were lots of doubts about the work in Korea, and they certainly falsified data relating to how these cloned embryos went on to develop. But they certainly managed to clone the embryos. What they didn't manage to do, and this was the claim they made, they didn't manage to establish a stem cell culture from those cloned embryos, and neither did they in Newcastle. It is difficult to achieve the creation of a human embryo by cloning, and it's proved even more difficult to use the stem cells or to establish a stem cell culture. So there's a kind of tiny step being taken there. And the question all this raises, I think, we are using human embryos here, and they may have been produced by IVF, or they may have been produced by cloning. But the question arises as to whether, if you like, our general motivation to heal, or our Christian mandate to heal, our Christian mandate to follow in the footsteps of Jesus to heal people, allow embryos to be the sources of stem cells. I talked at length with Susie Leather before she stepped down from the HFEA at dinner one evening. Her overriding motivation was that Jesus was a healer. This was a way of increasing the range of healing therapies that were possible, and therefore it was okay to go ahead. That was her take on it. But it raises all sorts of questions about the status of the early embryo, and so we need to pause to think about, what do we think humans are, and whether, at what stage in the beginning of life do we start to ascribe human personhood to the developing embryo or fetus? It's a big question. So what are humans? We had a little bit of a hint this morning that humans were more than a bag of genes. But we can describe humans at many different levels. We can first describe us in terms of chemistry, and I'm going to hark back here to Rodney's talk and I guess other people's talks. But this is what we are. We are stardust, we are golden. We are billion-year-old carbon. Joni Mitchell, the Canadian folk singer, must have a physicist friend, I think, because as Rodney explained to us, the elements that are bigger than helium have to be made in stars and are released by exploding supernova. And so it is absolutely true that almost every element in our body was made of a star. Joni Mitchell wrote that as the chorus of a song she wrote for Crosby, Stills, Nash and Young before Neil Young left them for the Woodstock Festival in 1969. Interestingly, it's been re-released, but with some of the science missing from the chorus. Dumbed down. Actually, it was interesting they kept the last line of the chorus, which says we've got to get back into the garden. So maybe there was a kind of anti-science, pro-religion take on the cover. So she knew her science, Joni Mitchell. But biology, again, as I said this morning, I have no trouble in identifying humankind as part of the whole overarching environmental system, because we are clearly mammals. There's no doubt about it, we're mammals. And if you look at a mouse genome, we're about 300 genes different. Now, 300 genes is actually a lot because of the interactions between genes. It's a lot of genes, but nevertheless, we can see in mouse exact parallels to things that happen in ourselves. And we can go back down the scale. We share about 60% of our genes with the haddock. And if ever you thought some of your friends have pond life, then we share about 40% of our genes with an average plant. And these are the genes that encode essential functions for cellular housekeeping. And so the nearer we get to us, obviously the others begin to fall away. So we're clearly mammals, and we're primates, and we're great apes and all that. But of course, there's more to it than just the straightforward biology, because we are the most complex of living organisms. Even people like Steven Pinker, who want to place us quite close to the apes, will point this out to us, that our next nearest relative is an endangered species living in a threatened habitat, able to modify its habitat only to a limited extent, as humankind has conquered every habitat in the world, and is in some ways over-dominating the world. We have self-consciousness. Now, we're not the only creatures with self-consciousness. The ability to recognize yourself in the mirror almost certainly is possessed by dolphins. I don't know how many mirrors there are down there, and certainly by elephants, and certainly by chimpanzees, and perhaps even by dogs. Although somebody said to me, dogs are self-conscious, but they're not conscious of that. I'm not quite sure what that means. This self-awareness, but it has a very high level in ourselves. We ask ourselves, who am I? And then we have rationality, we have intellect, we have moral reasoning, which goes far beyond the altruism that we see in animal groups. And then we can ask, what about high levels of existence, spirituality? And depending on your religious belief, you might simply talk rather vaguely that there's a spiritual dimension. And if you go down to the average bookshop, you may find a whole section on spirituality. And many people with no religious belief at all talk about spirituality. A pantheist might say that, a pagan might say we're one with nature, a pantheist might say we're part of the divine. Some people talk about the spark of God, but as Christians we're much more inclined to say made in the image of God, able to relate to God and with a number of the qualities that God has, which is very, very humbling and very scary in some ways. But we have a spiritual level of existence, and all those go together from the chemical right the way through to the spiritual to make up our humanity, what humankind is. And so we come back then to at what stage at the beginning of life, and we could also ask the same question at the end of life, what stage at the beginning of life do you want to ascribe human personhood? I personally find the question, when does human life begin, irrelevant. Human life begins, well, the zygote is human life. The question is, at what stage in that early life do we want to say that this is a human person? And here, this is where there's a whole range of views, possibly even in this room. But the two extremes of the spectrum are that the early embryo is a human person. And by the early embryo, I mean the one-cell embryo, the zygote, that we can't not call that a human person, because it's a unique genotype, unless, of course, it's going to have an identical twin at some point, and it's going to develop into a born human. At the other end of the scale, the human embryo is not a human person, and indeed, in some cultures, the fetus does not become a human person until it is born. And in terms of human rights legislation, the fetus has no human rights until it is actually born. And some cultures reflect that in the way that they treat the early embryo. So even though one recognizes that the early embryo will become a human, this school would not ascribe human personhood to that early embryo. and you'll find differences along the gradualist school about where you start to think about the early embryo or the fetus as a person. Should we attach significance in implantation? Should we attach significance at the first sign of the brain at about 4 weeks? Should we attach significance at the time when viability is possible at 20, 23, 24 weeks? Sentience, right? So there's a kind of a range of views here. But obviously, if you hold that view, then any stems and research on embryonic stem cells has to go out the window. And of course, our worldview may well affect our views. But I want to make it very clear here that as we look at the range of views amongst Christians, there isn't a clear cut, although the Glorious Christian Fellowship would have you believe that all Christians believe the one cell embryo is a human being. That is not so. There are many Christians who support research on embryonic stem cells. And there are also many non-Christians who don't. Many people with no religious belief who are very uneasy about using embryos to make stem cells. Come on. And just surveying one or two other faiths, Jewish believers on the whole are really happy with it. And in Islam, again, there's a range. It does depend which branch of Islam you hold to, but the view we most commonly encounter in Islam in Britain is that there's support for stem cell research, just as there is for genetic engineering of crops. And there's also this other question that I think will concern all of us who have a particular ethical take on what goes on in our name in the world around us. What do we do when our private morality clashes with the publicly accepted morality? So in the States you have pro-life groups bombing abortion clinics because as a protest that their private morality is being transcended by the public morality that allows abortion. So what sort of action do you take if you find yourself in the other camp, if society's gone one way or another? Okay, so that's the scenario that stem cell research is going ahead in this country. In order for it to happen, there had to be a change in Act of Parliament, which I'm going to talk about again in a few minutes. But the Human Fertilisation Embryology Act, as originally framed in 1990, did not allow research on stem cells. The only research it allowed on embryos was research that was related to human fertility and human heritable disease. So an Act of Parliament was needed in order to allow stem cell research of any sort, whether by cloning or by IVF embryos. And this led Bob Winston, who is quite an outspoken critic of government policies, believing that actually Parliament had been persuaded that this was on because scientists had actually distorted and exaggerated the potential of stem cells in human therapy. The idea that it was going to be the panacea for repairing damaged cells and tissues. And what he said here, I think, was quite right, that in the vote in Parliament in 2001, parliamentarians were persuaded that stem cell research was on the verge of such a major breakthrough that it would be a matter of a very short time before clinical treatments became available. We're going to look at that in a moment. So whatever your ethical take is on embryonic stem cells, whether you're okay about it or not, nevertheless, it seemed to me that an exaggerated case had been made. And this is Bob's view. He views it with growing suspicion. I will say he's actually jumped on the bandwagon since then. So that was quoting from Bob Winston's speech as president of the British Association in 2005, and Tim Radcliffe reported on it. Okay, so that's embryonic stem cells. I'm going to leave them for a minute. We're going to come back to them later. But it's not just embryos that have stem cells, because all the time, from fetal life onwards, there's a need to replace and repair cells. Now, in some parts of our body, that repair and replacement is actually extremely difficult and sometimes doesn't happen. So some parts of the brain become more or less irreparable, although there are central nervous system stem cells. But if you think about our bone marrow is producing, as we speak, millions of cells per minute. The red cell population particularly is in a state of rapid turnover. Each red cell lasts about 2 weeks, and so there has to be a constant turnover. So in the marrow of our long bones, we've got this block of stem cells whose function is to produce blood cells of all the different types, 13 or so different sorts of blood cells. And so they're replacing cells which are turning over. When we are injured or wounded, we have a repair system that calls into being stem cells to repair those things. When muscles are damaged, there are stem cells that replace damaged muscle, although I know for a fact as an aging athlete that they're harder and harder to call into action the older you get. But nevertheless, the bone marrow is still chuntering away there. And of course, their role is very specific. Their role is to function to produce cells of a small number of types, not the 210 to 120 in the whole human body, but a handful of cell types. In the case of the bone marrow, about 13. In the case of muscle stem cells, rather fewer. Limited potential in the human body. And people had wondered whether adult stem cells might be better than embryonic stem cells in this regenerative medicine, because you've got this thing in nature that they have a limited potential, so maybe they'll have a limited potential if we use them in medicine. Could you envision using bone marrow cells to repair muscle, for example? That doesn't seem logical. That's not what they do. But it had been suggested, and the Brain Research Centre here in Cambridge was back at the forefront of some of this, that maybe you can persuade these adult stem cells, I should say really call them post-embryonic or non-embryonic, because they occur from fetal life onwards, persuade these adult stem cells to go down a different pathway to what they normally go down. So instead of these bone marrow cells going nicely in to make the blood types, maybe we can make them make nervous tissue. Maybe we can make them make muscle tissue. Trans across differentiation, the pattern of development of individual cell types. Can we make them trans-differentiate? The answer is yes. And indeed, so far, the only clinical trials, apart from using bone marrows in bone marrow transplants for people with things like leukemia, the only clinical trials with stem cells have been with adult stem cells, and nearly all bone marrow. And there have been a small number of clinical trials looking to see whether bone marrow stem cells could repair other tissues. The success has been very variable. That a man who had severe heart disease and could not walk up his stairs without pausing to take breath every 2 or 3 steps had an infusion of bone marrow stem cells into his damaged heart. They've trans-differentiated into muscle. Exactly how that happened and the growth factors involved is not at all clear. On the basis of that and a small number of other cases, there are clinical trials going on to see whether heart can be repaired with adult stem cells. The success rate is low, we need to say that there have been some positive indications, but the overall success rate is very low, a small percentage. So clinical trials are being undertaken, but exactly how they'll proceed is hard to know. And then we've got a whole set of kind of off-the-wall research coming in from different labs interested in developments. So this came from a lab in Germany in 2006, where stem cells from testes, and what they do is they take them at a late stage in the formation of sperm, just before the division that produces the final sperm cell, just a couple of steps before then. And they're sort of stem cells. And it's been possible to make cells from those testes cells that look like embryonic stem cells. And when they're placed in a mouse embryo, they behave like embryonic stem cells. So we've got a situation here where stem cells have been generated from germinal tissue before the formation of the final gametes. And this is is where guys cross their legs, I guess. The professor of anatomy at Sheffield said that the biopsy was not that uncomfortable. and in terms of the immunological types across the UK, he reckoned it would take the establishment of about 150 different cultures to get a range of immunological types if we could repeat that. But this has not so far had any clinical application. Now, stem cells occur in all sorts of other places. They occur in the umbilical cord, and that's often used to try to replace damaged bone marrow in pre-existing children. They occur in the placenta, and in October 2006, the Newcastle group succeeded in inducing the formation of liver-like cells from stem cells in the umbilical cord. And a kind of tiny piece of a sort of liver-type tissue, about the size of a penny piece, was made. So there's an interesting example of another source of stem cells. This keeps jamming. and embryonic stem cells, well, we can get them now, it seems, from the amniotic fluids. And so you might be able to culture those up. And the question then is that if we got these other sources of stem cells, particularly the adult or post-embryonic, would that cut out the need for the use of embryonic stem cells? Will it ever do that? And obviously, if you're unhappy about the use of embryos to make stem cells, then you really want to push that. And there's an argument in that school, in that group of people who are opposed to embryonic stem cells, suggesting that too much money and effort and publicity has been devoted to embryonic stem cells rather than adult stem cells. Okay, well, let's look at some recent developments over the last year. Bone marrow cells. Can we make them transdifferentiate? Well, here in the lab, a heart valve was grown from bone marrow stem cells. They're clearly transdifferentiated. And then, this is, I mean, I was surprised at The Guardian having scientists in the sperm cell breakthrough, but that's sort of more of a kind of Daily Mail headline. But nevertheless, what they did was they took bone marrow stem cells and persuaded them to go down the route as if they were germinal cells and made immature human sperm. So these bone marrow cells, at least in the lab, seem to be able to be transdifferentiated at will. And then we had whole blood vessels being grown on a little kind of biodegradable former to make a tube from bone marrow cells. So in the lab, at least, bone marrow cells are doing very well, even if the clinical trials have had limited success. Now, what's all this about? I quickly passed over the need in cloning, making embryos by cloning, the need for an egg that has to be emptied of its DNA. But those eggs have to come from somewhere. And egg donation, as we heard in the little video this morning, is not a comfortable process. It's rather harder to donate eggs than it is to donate sperm. And you'll need a lot of very altruistic women to contribute to this, and maybe their own egg supply is limited. There were ethical issues arose in Korea when young female researchers in Professor Huang's lab were persuaded to donate ova for the research, which would be completely unethical in this country. And so it was suggested that we might use empty eggs from other mammals and use them as receptacles for the human genetic material, not forgetting the mitochondrial DNA that our colleague mentioned earlier, but that's another question. We might empty the egg of its main genetic material, and we might grow human stem cells, if you like, using the empty mammalian egg as a kind of receptacle. And so application was made for human rabbit. Of course, they're not really human rabbit embryos. They're empty rabbit eggs with human cells, human DNA, and then the human cow. So both these came up in the autumn of last year, about this sort of time. And then the HFEA met and considered it, and on January 11th, they said, no, we need to think about this more. This is actually quite an amusing thing. I was giving a talk in Southampton on the evening of January 11th, and and I knew that the decision was being made at 2 o'clock that afternoon. So as soon as I got to Southampton, I got on the wireless broadband and checked out the HFE website and was able to give them the decision there and then at the meeting. So they said no. We don't know enough about this, we don't know if it's safe, we don't know what the public views are, we want time to think about it. The idea that we use these empty, non-human mammalian eggs to receive human DNA. But we live in a political world. I've already said that the use of human embryos in stem cell research required a change in the Act of Parliament. And so in 2001 that was driven through. It was driven through partly by the advocacy of Dr. Ian Gibson, a former molecular biologist who's now an MP for Norwich North, and by Dr. Evan Harris, Liberal Democrat spokesman on health affairs, who's a former doctor in Oxford. And those were very eloquent in pushing this through. It was in a sense, a political decision. And Ian Gibson said to me just before the vote, he said, Britain has got a lead in this research. We must keep that lead. It's important that this Act of Parliament goes through. And so the MPs started to say that what the HFA had said in January 2007 was not acceptable to them. That they shouldn't hold up this research by having a year's worth of consultations. And they overrode the HFEA in May this year, before the HFA had completed its consideration, and said, you will allow it. So we live in a political world. Whatever you think about the acceptability of the protocol, it seems to me that political expediency is winning over considered thought. And one more thing to think about. As I've said, there are clinical trials in progress with adult stem cells, particularly in relation to the repair of damaged heart tissue. And there are good reasons for supposing that other clinical trials will ensue from the basis of lab work. But despite the extensive effort that is being placed on embryonic stem cells, particularly in Stephen Minger's lab, and he's done a really wonderful job of demonstrating that different sorts of cells can be made from embryonic stem cells. No clinical trials are underway yet. And the scientists are talking about official indecision. What they actually mean by that is that no health authority at the moment is willing to give the go-ahead to any form of clinical trial of embryonic stem cells because of the worry about being sued if they go wrong. There's a kind of anxious state that there may be litigation if the treatment goes wrong. So no regional area health authority will say yes at the moment to clinical trials with these embryonic stem cells. And once again, I'll advertise the weekend in Devon because we're going to be dealing with that as well. And I'll stop at that point. Thank you.
Thank you very much indeed, John.
I need to just check my medical facts here, correct me if I'm wrong, but my understanding is that when a person has their heart repaired, the heart is taken out and put on a heart-lung machine while they do repair. And then when the operation is completed, I've heard it said that the person often undergoes a personality change. Is that correct?
The heart is usually repaired in situ actually without taking it out, but the heart function is bypassed by the machine whilst the repair is going on. I've only heard that said in relation to people who have received a transplanted heart, but I haven't seen any hard and fast data on that, so I can't tell you whether that is so or not.
Okay. Well, let's just assume for a minute that it's reasonably true, because it raises the question of, does a cell need to perform naturally in its context? If you take the cell out of its living context and put it in a lab situation, it changes the relationship that cell has with that living contextual environment. And I'm quoting the heart case to put a question mark around whether the cell or the tissues or the organ then change in some way?
I don't know. And one thing I will say, if you have a transplanted organ of any sort, you're taking into your body, obviously, a whole chunk of cells that have got somebody else's genotype in you. And maybe that will have an effect on you. But I just don't know enough about what you're saying here to make any sensible comment. I don't know if anybody else has. It's nothing I've heard of that an individual cell. I mean, obviously, epigenetic effects will affect the way that genes are expressed, but that an individual cell can, in some way, take that back and change the way a person is. I've not come across that at all. It seems to me to be way out on the edge.
Thank you. More questions?
Stunned silence. It's late in the day.
Yeah.
When you were talking about the totipotent zygote, a thought flashed through my mind which may be totally irrelevant, in which case you'll tell me. And it suddenly occurred to me that that there might be an analogy to be drawn from that to the totipotence of the original Big Bang particle, which contained in it the totipotence of everything that's followed since. Is that a meaningful or a stupid remark?
It's a different use of the word, but it's quite relevant. I mean, I don't think we can sort of parallel the life of individual human with the life of the universe, except to say that they're in God's hands.
Interesting thought.
Absolutely.
That's rookie.
Yeah, I'm not sure what I want to quite say about that. Good. I mean, that raises far too many questions, I think.
Yeah.
Not about your topics.
You were quite right to point out that in the United States, there is no ban on using embryos. There is a ban on federal money, on tax dollars supporting it for a whole host of political and ethical reasons. I've heard it said that the private sector is not enamored of embryonic stem cells. They're investing in adult stem cells. They don't see, and as we all know, they tend to be profit-oriented, and they don't see that there's great value in going down that route, whereas adult stem cells are very useful and are profitable in some sense. It's obviously an immature technology, if you will, so they're working on it. What are your thoughts on that?
Well, I mean, firstly, adult stem cells is not immature technology when it comes to bone marrow transplants, and so there's money in that. It is true that that the nearer you are to the dollar, the more active is the research line. But there are companies and charities in the States who are working on embryonic stem cells. If you Google that, you'll find them. They're mostly going by the cloning route, trying to. I suppose we should call it somatic cell nuclear transfer, which gets out of the use of the word cloning. But there are companies over there. I mean, I should have brought a list with me, but I found quite a few the other day.
But is the reluctance perhaps to do with the different climate in the States about the ethics of stem cell research, of embryonic stem cell research?
No, I don't think so. I mean, if a Maury poll conducted in the States about 3 years ago suggested that about 70 to 75 people, and it was more or less evenly divided between the two sexes, were positively supportive of the idea of embryonic stem cell research, which means that some of those must have been evangelical Christians. So 18% were dead against, and the rest didn't know. And about the same proportion in this country. The data were very similar. So I think the population at large is actually much more supportive of the research than we would get the impression of reading the paper. You get the impression of all these right-wing evangelicals are persuading the president that he mustn't fund this stuff. But there are clearly evangelical Christians who are completely happy with the idea, and other people as well. 70-odd% would support the work.
Yes, quite where polls get you on this. I'm not
No,
sure.
but yeah, okay. I mean,
you've got 50% of creationists.
But the English, the British one, which was commissioned by Wellcome, was actually fascinating because they took 1,015 people and just asked the questions. They took another 1,000 and showed them pictures of the early embryo before they asked the questions. And the latter group were even more supportive of embryonic stem cell research.
Yeah.
One of the worries I would have is perhaps less an ethical one as possibly a scientific one, on the grounds that it seems to me at least possible that a number of cancers might easily be being produced by effectively turned off genes being turned on when they're not supposed to, and therefore that to introduce DNA into a person that has that kind of potential could easily produce a kind of cancer time bomb.
I think this is particularly true of cloning or somatic cell nuclear transfer, where we know so little about the epigenetic effects on, if you like, the quality or the functioning of the DNA that accumulate during a person's lifetime. We know for a start that the ends of chromosomes, they're called telomeres and they stop the chromosomes fraying, they decrease in length with age. Now, can the embryo repair those? Can it lengthen them back to embryonic state? We also know that as we get older, our DNA repair mechanisms, and where our DNA is being damaged constantly and it's being repaired constantly, our DNA repair mechanisms become less efficient, and so those could be carried through in a nuclear transfer. And it seems to be quite possible that some sort of epigenesis may occur in reactivating oncogenes, which should only be used at particular times of development. So I think I share that concern, and I share it particularly if we start transferring human nuclei into the empty eggs of other mammals. And so even though I can't see any problem with that research program, I don't think that such cells should ever be used in human therapy, because it's a complete unknown as to what the epigenetic imprinting might be from the receptive egg cytoplasm and from the mitochondrial DNA of the receiving. So I think research is, for me, that research is okay, but as a therapy, I would avoid it, I think.
Interesting, yes.
Kind of following on from that, is that what you were implying when you said, I think you were saying something like, Dolly died young almost as if the genes had developed 6 years ago?
Yeah, absolutely. That's exactly what I was implying. That 6-year-old sheep is relatively young, but it certainly wasn't a young juvenile sheep, and so the DNA repair mechanisms will have begun to decline. The telomeres at the end of the DNA will have begun to shorten, and so it may well be, that Dolly she suffered some respiratory condition, and she suffered arthritis, which is very unusual in a 6-year-old sheep, and it might well be that we were seeing there the effect of older DNA, which the embryonic system had not been able to repair fully. We just don't know.
Yeah, just to follow on from that, though, you then kind of implied that, I think you said something like, this is getting better.
Yeah, I don't know why, but as, for example, there's been quite a lot of cloning of domestic cats in the States. People are trying to replace their beloved pets. And it may well be that the technology is getting better as we find it easier to reinduce the embryonic state. And so there seems to be less and less. I will say for primates, the problems are only just starting. A number of people have been able to clone a primate. I think only, including 2, or maybe a few more, a tiny number of human clones and a tiny number of great ape clones. They're very difficult. And so all those difficulties have yet to be overcome.
Well, maybe I can throw in a fundamental one,
Yeah.
going back to the status of the embryo. We had this discussion yesterday about the firemen who went into the building not knowing whether there was anyone
That's
in the
right,
building.
that's right.
But on the kind of presupposition that there just might be, and then they lost their lives. I mean, I assume, are pretty, you've got to be pretty certain that you're not dealing with a human person in order to say yes to it.
I think if you're working with embryonic stem cells, you must be absolutely certain.
Yeah.
Otherwise, all the time you'll be wondering, am I killing somebody?
Exactly.
And
So it doesn't need that certainty.
You can't. If you talk to Stephen Ming or to Susie Leather, they're both convinced that you're not ending the life of a person
Yeah.
in obtaining stem cells for an IVF embryo. Mm-hmm.
Yeah.
So I guess if there's a fire, do you rescue the lab technician or the clone bank.
That's interesting. Yeah, do you want to go?
I was saying that this thing about the age of the DNA,
Yes.
presumably then that means there's some kind of resetting of the age of the DNA at the time of the producing of the sperm and egg.
When you produce the germ cells, the meiotic DNA replication, they produce, if you like, DNA which is germinal. The germline produces young DNA. But of course, the other question we need to ask is, what about those stem cells that are working throughout our life in the human body? Some of them remain active and produce DNA that will last at least the weeks that is needed for blood function. And why do some stem cell populations, if you like, become less active as we get older? For example, in muscle repair. There are stem cells in brain, but they're incredibly difficult to get to be active. Very, very hard indeed.
Yes.
Could you say a bit more about Jewish and Muslim attitudes? It was fascinating to say that there's a lot more of a consensus amongst.
There is. I mean, if you talk to Jewish believers, mostly, there's a very strong sense amongst Jewish believers that science is a gift from God, and that medicine is part of God's gift to us, as one of His created gifts, and that we should use it. In Jewish law, in Old Testament law, the developing fetus had much less protection than an already born human, and so that if you caused the miscarriage of a fetus, you could not be accused of murder. There were penalties to pay, but it was not to do with murder, and that goes through Jewish thinking today, that there's the moral status of the fetus is rather less strong than it is in some strands of Christian thinking. In Islam, they have this very, very curious idea that at 40 days of pregnancy, the fetus becomes morally significant. And why 40 days, I don't know. It's sort of almost Aristotelian.
That goes back to Aquinas too.
It goes back to
He taught
Aquinas.
that in the Christian tradition, didn't he? Yes. I mean, from a modern point of view, I think that's arbitrary.
It's totally arbitrary, yes.
Quite.
Did I read somewhere that it's different from a boy fetus from a girl?
That was Aristotle thought that.
Right.
Aristotle thought that the female fetuses became humans rather later than the male fetuses.
Now, Pauline has a question.
I just a question to do with what you just said. Is it actually criminal within the Christian West to cause a fetus to be miscarried?
Only if it's by an act of assault, because
Yeah.
otherwise abortion would not be allowed. So if you deliberately assault somebody in order to induce a miscarriage, that is a crime.
And it doesn't matter at what stage the pregnancy.
Oh, that's an interesting question.
I just wondered.
I'm not quite sure there have been enough cases for that to be tested.
Yeah, that's just a thought.
Yeah.
Your basic ethical distinction seems to be between human life and human personhood, and I'm wondering how that distinction is made. Is it the difference between the general and the specific, or what?
It really does depend, I think, on what of the range of qualities that I talked about. What range of qualities you need to ascribe personhood to the being. It becomes very, very interesting at the other end of life, when people begin, for example, through dementia or whatever, to lack capacity, and we wouldn't say that a person who had Alzheimer's and was in the late stage of dementia, we would be very hard pushed to say that they were no longer human beings. And so if we go back to the beginning of our lives, what attributes of that list do we need to have to make people humans? I mean, babies don't have moral reasoning. And Peter Singer, the very controversial Australian who is head of ethics at Princeton, would say that a newborn baby had less moral significance than a 3-year-old chimpanzee, because a baby does not exhibit all the things that we attribute to personhood.
And infanticide is
In fact,
okay.
yeah. So euthanasia of newborns is quite acceptable to Peter Singer.
Well, that's very disturbing. I mean, have you got a view yourself, John?
Have I got to view myself? Well, I mean, in terms of human development, I think firstly, there's a very significant change in the natural status of the embryo at the implantation stage. A relationship is formed. That relationship is really a physical and physiological one, but there is a relationship between the embryo and the mother. And until that relationship is formed, nothing can happen. I mean, Gareth Jones argues this quite strongly in one of his books, although I think he fudges the issue by saying that we can make the choice as to whether to give it moral status or not. Does that mean that I'm against abortion? No, I don't oppose abortion all the time, because I think sometimes it is the lesser of two evils. That that there are shades of grey here, and sometimes it's the less grey of the two.
Well, that's an interesting way of moral reasoning. Actually, just
Well, sometimes you have to do
I'll
that.
mention
Sometimes
yeah.
moral issues are not black and white.
Well, yes, which is the lesser of two evils.
Okay, well, the lesser, the more good of the two. This is very consequentialist.
Exactly. I mean, that's the point. That's my point. Yeah. Perhaps I will just throw in that we've got a course on ethics, which goes into bioethics. John's coming back, and we've got various other people, including a professional Christian ethicist.
Philip Taylor.
Philip Taylor, we've got Neil Messer,
Okay.
and various others.
You ought to get Donald
This is in April.
Mahuna from Dublin. I'll give you his name as well.
Oh, yeah, please do. I mean, that's just a kind of aside in the middle of John's question
But
time. To let you know, that's in April, and if these areas interest you.
I think we should also attach a lot of significance to viability as well.
Right. Okay.
Survival outside the mother.
Right.
So there are a number of steps that one goes as the fetus becomes more and more near and near birth. But it is true that we couldn't attribute full human personhood in terms of that range of qualities to a newborn, and yet we know they're a different person.
Yeah, exactly.
Viability
has effectively been rejected by the recent by Parliament because they failed to take into account those viability at 23 weeks or whatever.
Although the viability is very dubious in terms of the life of the surviving infant on the whole.
But why is viability important? I mean, I think I probably feel that it probably is emotionally, but I wouldn't be able to express it rationally.
Yeah, I think we're saying that if the little being can survive outside its mother, then we should treat it as if it was a born baby.
Right.
I think that's what we're saying.
I don't feel that my question's been adequately dealt with. Let me put it in another
I'm sorry about that.
On what basis then are you so sure that in doing this research on early embryos you are not destroying a human person?
Because I really do find it, and this is a view as a developmental biologist, I find it very, very hard indeed to ascribe personhood to a ball of cells, undifferentiated cells. That seems to me to be pushing the idea of personhood just too far, an undifferentiated ball of cells. And you may well disagree, but that's just where I stand. I will say, however, that I have some hesitation about using embryos for stem cell research for other reasons. I'm a bit worried about creating embryos just to make spare parts, because it seems to me that is a commodification, which may be a step too far, even though I can't ascribe human person to a ball of cells. But to make an embryo just to make spare parts of somebody who's got a bit of a problem seems, I just wonder whether that's appropriate or even right. So if you read Life in Our Hands, you'll see that we write that one of us is okay about this and one isn't. The one who's okay about it was actually the medic, my colleague John Searle.
And also ordained.
And also ordained, yes.
Yes.
But just for clarity on that point, your undifferentiated ball of cells is a pre-implantation
Embryo, yes, that's the point at which
pre-implantation ball of cells. Post implantation.
It doesn't remain a ball of cells very much longer.
No, and you would not carry out stem cell research.
Well, you can't.
You can't.
No, you can't. Once the embryo is implanted, you can't.
Ah, I see. It's physically impossible.
You actually.
So the moral question doesn't
well,
arise.
you're probing into the uterus to pull an embryo out. Well, you wouldn't do that.
Right. Okay.
At least I hope you wouldn't.
No, well, you tell me. Yes. So will you morally or practically?
Well,
or,
I think both.
Or practically.
I think both. I think this has now started down the development.
So that is your definition. It's implantation.
Implantation is a very important step for me in my moral reasoning.
Right. That's helpful. Thank you.
But as I said to Rodney just now, that doesn't mean to say that I'm totally opposed to abortion because.
No, I heard that too.
In terms of what we do with the embryos, you can't actually practically manipulate them when they are attached to the wall of the womb. You can only have them beforehand because they've started to go down that formation of the 3 layers of cells as fairly soon after they implant.
Fine. Thank you.
But the embryos you experiment on, are they ones that you form in a petri dish outside the womb?
Yes. Yes.
So you really can't then compare this idea of implantation because it doesn't occur.
Well, that's exactly what Gareth Jones would say. Because we don't allow implantation to occur because they're outside the womb, then their moral status is of a pre-implantation embryo. You see, you're going just like I did. You made that choice not to implant them. Yes.
Quite. I mean, that's a well-made point. Thank you.
Yeah.
And I'd also suggest that I came across that the United Nations had a clause that the fetus had to be protected before and after birth. And so I raised the question, how was it legal that Britain should have passed such an act? Because the United Nations formulated that clause long before the Abortion Act.
I think 2 things here. Firstly, the pre-implantation embryo is not a fetus.
No, I'm going back to abortion now, sorry.
Okay. The United Nations statement does not confer full human rights on the fetus. It specifically doesn't. So although it talks about a degree of protection being afforded, and that's really to protect the mother who's carrying the fetus, it does not ascribe, it does not allocate full human rights to the fetus. It specifically doesn't. And so abortion is not forbidden by the United Nations Act.
So I've misread the quote there.
It's all about
the Hippocratic Oath, then.
Yeah.
Well, it depends. Yeah, remember, doctors no longer swear
quite.
the Hippocratic Oath. This they agreed to the Declaration of Helsinki and Geneva post-war. It's what the doctors do now.
John, some don't even do that.
Some don't even do that. It's quite right. You're quite right.
As I see it, John, I wonder whether some of the conundrums arise because it's very difficult to draw a black and white line along the spectrum.
Yeah, thank you. I agree.
Both at the beginning of human life and the end of human life. And, I wonder whether drawing a line achieves too much anyway, because wherever you draw the line, it's to some extent going to be arbitrary. And whether one should rather say, look, it is human life, it's not personhood. I'd attribute moral responsibility to personhood, some sort of moral thing, as opposed to trying to define personhood around biology. But clearly there's a gradation along here, and how would you subscribe to the view that as we go further along that gradation, we should treat the individual with greater moral seriousness, rather than getting into drawing black and white lines?
I don't like black and white lines, except there are some biological black and white lines, and one is the completion of fertilization, at which point the embryo starts to develop, and the second black and white line is implantation. Those are very clear events. But after that, it is, if you like, a spectrum of increasing developmental complexity. It's a gradualist position. And that's the position I really hold, that, at no point can you suddenly go past another clear black and white line.
So the primitive streak wouldn't be one?
No, the primitive streak is simply an indication that a nervous system is going to develop, and that's actually quite soon.
But that was the one the HFP...
Well, the HFP, but that was, remember, that was an in vitro This was embryos in vitro, and the idea was that they didn't want to allow embryos to develop too far before they stopped allowing research. And the primitive streak is, if you like, an indicator sign that we're beginning to see polarity. We've certainly got a front and a back by now, and we're beginning to see where the central nervous system will, in about 3 weeks' time, start to be laid down. The earliest visible central nervous system is about 4 weeks. The primitive streak is an indication of that's going to happen.
Thank you.
Just to say, my father died of Alzheimer's disease, and one way of defining Alzheimer's is to see it as a gradual deterioration of the personality. And in the latter stages, he was clearly not a person in the sense that we knew.
Absolutely, as I was hinting earlier, yeah.
And I mean, any decisions, let alone moral decisions, had to be made for him by the next of kin.
Yeah.
And I wonder whether, by analogy, you could argue at the other end as well that the development of life forms along the scale we need to treat with increasing moral seriousness, and that, as it were, the kin needs to advocate appropriate care so that you don't just treat them as commodities or treat them as some sort of piece of garbage. I mean, they are human life, but they're not personhood. And so the kind of the moral rules are, it's not as though you've got a set of morals for a person and a moral set of rules for how you treat your car, and there's nothing in between. But in actual fact, there are a whole series of grey areas here that drawing a line actually doesn't necessarily help.
Well, I think I'm with you there. One is unpopular in certain Christian circles if you say that it's very hard to be black and white. But I am also a gradualist in this. But it's very hard to write a law, if you're interested in the law of the land, based on gradualism. And for example, at the beginning of born life, parents make decisions on behalf of their children. In law in Britain, that goes on for quite some time, at least until they're 12, maybe longer, depending on the circumstances. In the Netherlands, incidentally, and in Belgium, a 13-year-old has the right to opt for euthanasia, which is interesting. So, I mean, we do in fact work like that in the law once the child is born, that we say up until a certain age, the parents are responsible for, and after that, and in different areas, it's different ages. In medical treatment of consent, legal age of consent for sex, and so on. It's different steps.
So I understand one of the basic ethical rules for general practice is you should not intentionally do harm. You know, the kind of
yeah, that's one of the Childress and Beecham 4 pillars of medical ethics.
Right. And so if you applied that to the whole area of the stem cell research, the argument would be that if you are going to do some harm to this form of life, that there are clear ethical benefits that would justify that harm being done.
Indeed, you see, one of the other 4 pillars is beneficence, that you seek positively to do good. So if you have a patient who would benefit from a stem cell transplant, then doing good to the patient will result in you having to use cells derived from a human embryo.
So you would connect that. You could connect that kind of utilitarian approach to your colleague's thing about Jesus was a healer. That if you were doing this for the benefit of a healing outcome, then you could justify it, and you would have to do this on a case-by-case basis.
Well, I think my friend Susie would say that. That, you know, there are some areas where the use of stem cells would be trivial. Just because somebody doesn't want to get old, when it's just the normal process of old age, might well not be a case where you consider it the good outweighs the harm. If some person has been badly damaged by a trauma or accident, and you had the possibility to repair them, then the good may outweigh the harm. So it is almost a case-by-case basis. But remember that one embryo can give rise to a lot of stem cells because of the possibility of culture in a new lab.
It's reached 1:30, but maybe just one last question. Thanks.
Just thinking of something you hinted at again a bit earlier about stopping before we get too far. It just gave me the image of if you're going down a very icy hill, you've got to decide to stop.
On a black run on your skis, yes.
Yeah. Because you don't want to be in the pond at the bottom. But the point is, unless you stop at the top, that's where you're going to end up.
Yeah, well, I think there are slippery slopes and slippery slopes, and I think on the whole, although I think there are some which are clearly slippery slopes, I think in this area we're not in a slippery slope. We're in a ring fence situation. You go up to the fence and say, should I climb it? So, and then that might make it easier to climb the next fence. But I don't think we're on a slippery slope here. I think we're definitely in a series of decisions that have to be made.
So I was thinking, like with abortion, that whereas you start with a very clear cut, it is for the health of the mother, to the point where I have people say, women have said to me how, having gone to the doctor to say, am I pregnant? And the doctor said, yes you are, do you want an abortion? You know, without them having even mentioned it.
Yeah, that's a slippery slope.
Okay, well, I think we'd better stop and give John a big round of applause. Thank you very much.